So my name is Mojgan Rabiey. I'm from University of Warwick. And in my group, we use bacteriophage to control important crop diseases. So one is bacterial canker of cherry. So cherry canker is a complex disease. It's caused by Pseudomonas Syringae pathovar. So we have pathovar Syringae and pathovar morosplenorum, and there are different races. So this phylogeny tree that is from isolates that we recently collected from cherry orchards around the UK, and we found some new isolates. But what I wanted to show here that PSS and PSM race 1 and race 2, they belong to different phylogroups, and each one have their different genetic diversity and with different pathogenicity and virulence, and therefore that's why we call this a complex disease. So this is a pathogen that could infect all parts of the plants. So it could infect the leaves and making the shot holes. It could infect flowers and then necrosis on the fruit, and then huge amount of oozing and bleeding from the trunk of the tree that it seems trees bleed to death. So UK produces around 12,000 tons of cherry each year. That is worth 34 million pounds. But this disease is quite devastating for the farmers because it could, if the cherry orchard is infected, it could infect up to 75% of the trees. So there's no current effective control measures. We can't use copper anymore because of the legislations, environmental impact, bacterial resistance, and breeding for resistance for trees is quite time-consuming, expensive, but also because it's a complex disease. There are different pathovars of the pathogen, and therefore making breeding difficult. So the only method currently is that for the cherry farmers, they cut down the trees entirely. So that's quite devastating. So we want to use bacteriophage or phage or viruses that infect and kill bacteria. So they are very host specific, and so they only infect the host they come from. So how do they infect? So they bind to the bacterial cell surface or receptors, and they inject their genomic material inside. They take over the replication machinery of the bacterial cells. They make their own particles, and they lyse and burst the cells open. And each bacteriophage goes and infects the neighboring cells. So that's the beauty of using phages, because one phage infects one bacterial cell at the same time produces hundreds of other phages that they could infect the other bacterial cells. So for this purpose, we went to different cherry orchards around the UK. We collected soil, leaf, bugs. We isolated lots of phages, characterized them, know the efficacy in the lab. So we know the temperature stability. We know the genome sequence them. So we know the receptors they use on the bacterial cell surface. We also know a lot about our PSS and PSM populations and the antiphage defense system and how we could use those phages against, and we do a lot of host range assays. So as I said, we isolated lots of PSS and PSMs from our cherry orchards around the UK. And here I'm showing you the 5 different phages that we tested against hundreds of these PSS and PSMs. And most likely you can see that these phages have different host range, but mostly they could kill more than 60% of the panels that we tested here. So we also test, we do a lot of glass house assays that we test the efficacy of phages to reduce the symptoms and bacteria population. So here we tested the PSS and 5 of these phages individually and in cocktail or mixture of 5. And we infect these woody tissues, and we score the disease, and we also do bacterial re-isolation. We count the population to see the impact on bacterial population as well. So here you can see when we infect the cherry woody tissues with the pathogen, Pseudomonas syringae, so you can see this browning lesion on the trunk or the woody tissues. When we have one phage, it reduces the symptoms. When we have the cocktail of 5 phages, clear the symptoms compared to negative. And when we count the colony-forming unit or bacterial population, we can see compared to the control, the phages could reduce the bacterial population significantly. We also done the first UK phage field trial and we went to the field not knowing what to expect, and we wanted to, so we sprayed the leaf and woody tissues with bacteriophage, with pathogen first and with the bacteriophage, and we wanted to understand how long, for example, phages could stay viable and effective in the field. So we found that, for example, our phages could survive up to 30 days when the host is present. We found that the phages don't impact the bacteria, and therefore no resistance emerged to our phage application. We found that the bacteriophages don't impact the bacteria, the fitness, for example, the growth and pathogenicity and virulence. And also they don't impact the microbial community, like fungal and bacterial community. So it was a successful field trial for us. But knowing all this that I showed so far, for bacterial canker of cherry, one of the most important stages of the disease is the flowering stage, because that impacts the fruit production. Therefore, like to have a phage application that is successful, we need to deliver phages at the right time, at the right place, and in a cost-effective manner. So the producer of commercial fruit or cherry farmers, they always have bumblebees in the farm. They use that to enhance pollination and maximize yield. So this is the existing and routine agricultural practices. So as I said, we do a lot of sampling. So in one of the sampling, I thought like, why not to use bumblebees to deliver phage flowers, because it's very difficult to a flowering stage, because the flowers open at a different time, and by spraying we are not getting the phages to all flowers, so to protect them from bacterial canker. So we wanted to use to see if we could use bumblebees to deliver phage from hive to flowers, from flowers to flowers, but also by doing so they could reduce the bacterial population. So I'm focusing on this, if bees could transfer phage from hive to flowers and reducing the bacterial population. So we use two different systems. So we use artificial flowers to mimic the cherry flowers, and we use also real cherry flowers with trees. So we made our own phage powder using our in-house protocol, and we placed the phage powder at the hive entrance, so bees had to go through the phage powder before leaving the hive, and then we removed the phage powder at day 1. So it means that bees only had access to powder once. So we did the same for cherry flowers, and we collected samples at different time points to understand. But we also tested before this to understand if the phage powder is stable at different, for example, on leaves and the feed that we created, and so on and so forth. So here I'm showing you the percentage of flowers positive for phage. So as I said, bees only had access to phage at time 1. So time 0, that if we just check there's no native phage on the flowers. And then time 1, you could see 50% of the artificial flowers and cherry flowers had bacteriophages at 10 to the 2 to 10 to the 5 platform unit per mil. So it proved that bees having access to phage once, they could move the phages to flowers. So then we wanted to see if by doing so they could reduce the bacterial population. So again, we have the artificial flower setting and cherry flower settings. So this time we infected the flowers with pathogens, so in both settings. And we have different compartments. One that the bees don't have access to the phage powder, and when they have access to the phage powder, again, we remove the phage powder at day 1. So it means bees access to the phage powder only once. And then again, we collect the samples at different time points. So again, so you can see at time 1, bees could transfer phage from hive to flowers. And by doing so, they reduced the bacterial population significantly throughout the experiments, and we couldn't detect any at time 7 when there was a phage present. And the real cherry flowers, they again reduced the bacterial population throughout. So it means that by only having access to phage powder once, they could reduce the bacterial population throughout. whether we could, it might be that we couldn't just detect the phages by the spot assay that we do, and we need to do more like qPCR and so on to detect phages here. But having said that, working with trees is quite challenging. For example, the number of trees that we could use in the glasshouse, the number of flowers that each trees have. So for example, at the artificial flowers, we had like collect 24 flowers per time point, but here we only had access to 12 flowers per time point. So there's a difference in terms of number of flowers we've taken. Also, like there's a short window that we could do the experiments when we do the real cherry flower experiments. So we showed that, like, yes, bees could transfer phage from hive to flowers and could reduce the bacterial population. But we also collected the bees' body at the end of the experiments, and we found that when bees had access to phage, there was no pathogen on their body. So it means that by using phage, we could also disrupt the cycle of pathogen transmission between flowers to flowers. So as I said, this is the existing agricultural practice that we should be using to control this important crop disease at the right time. So another system I just want to mention quickly that we work on is the black rot of Brassicas. UK produces around 300,000 tons of them, worth around 400 million pounds. And you can see the symptoms on vegetable Brassicas. So it's caused by Xanthomonas campestri, or XCC. So you can see this V-shaped symptoms on the leaves. And when this happens to a farm, farmers need to remove the infected plants one by one. So it's quite time-consuming, expensive, and sometimes when the infection is high, farmers need to abandon their farm because it's just not manageable to use. So there's no current effective control measures to control this disease. So at Warwick, we sequenced around 500 different XCC isolates. So you could see here that we have more than 9 races. So again, this is a very complex disease. So there are different races with different genetic diversity, different pathogenicity, virulence. That's why controlling this disease is also difficult. So we've been collaborating with Agri, having access to soil and leaf samples from the farm. We have managed to isolate lots of phages. So here there's a panel of 6 of those phages that we isolated, that we tested against 150 of those XCC that we recently sequenced. And you can see these phages have different host range, but they could kill more than 60% of these XCC that we tested so far. We're doing more tests as we go along. Well, I just wanted to highlight this so you could, Sophie Wright, the summer student supported by AAB, so she's having a poster on this topic, and so please have a chat with her and visit her poster. So in that note, I want to thank all the collaborators and funding bodies, and thank you all for listening.